Okay, we can go ahead and get started. It is my pleasure to introduce Dr. Nicola Allen today. She received her PhD at the University College London before coming to the U.S. At Stanford, where she completed a postdoctoral fellowship. And she worked with Dr. Ben Barres, who is someone who was known to quite a few of us. She joined the faculty at UCSD in the Department of Biology in 2013, and she continues as faculty in that department, but has since joined the faculty at the Salk Institute of Biological Studies. The research focus of Dr. Allen's lab is the role of astrocyte neuron interactions in health and disease, with a particular emphasis on identifying molecular mechanism astrocytes used to regulate neuronal synapses. The long-term goal of her research is to use astrocyte-derived signals to stimulate synaptic repair in disorders where synapses are lost. Dr. Allen has published widely with numerous articles in top-tier journals, including Neuron, and publications over the years, and a very large number of publications. She is also, last but not least, she is a dedicated mentor of students, including PhD and postdoctoral students. The title for the talk today is Using Astrocytes to Target Dysfunctional Synapses in Alzheimer's Disease. Welcome. So happy that you could join us. Chat. I don't know if the problem. Oh. Do people have a problem with the audio? No, someone did. Everyone could hear. Okay, great.
Well, thank you for the introduction and for the invitation to be here today. And it's really great to be giving this local talk so that I can share with you what my lab is doing at the Salk Institute, now moving into the area of Alzheimer's disease and neurodegeneration. And hopefully what I present today can stimulate some collaborations in the future and future discussions. So my talk is up into 3 different sections. The first part, I'm going to spend about a third of the time going over why we're interested in astrocytes, this cell type that you see here on the screen, this one cell shown here, what they do in the healthy brain in the typical situation during development, which is where my lab previously really focused, understanding astrocytes in the developing brain, and then lead you into how this knowledge that we've garnered of how astrocytes regulate neurons in development, we think could actually be applicable to contexts of neurodegeneration and Alzheimer's disease. So in the middle third of my talk, I'm going to tell you about some work that is unpublished but recently posted on bioRxiv if you're interested, where we've completed a study looking at how astrocytes change across Alzheimer's disease disease course and ways that we can rescue some of the dysfunction. And at the end, I'll quickly go through some of the other projects we have in the lab to give you kind of a flavor of the different approaches that we're taking to give you an idea of potential collaborations, as I said, that we could make. So before I get into the details of what I want to talk about, I realized that we have a diverse audience of people thinking about neuroscience in the brain at many different levels. And just to orient you, what I'm going to be talking about mostly today is things at the cellular and molecular level. So we can think about studying the cells that compose the brain, so the billions of neurons and glial cells. And what I will mainly talk about today is thinking about how those neurons communicate through these trillions of different synapses that are formed during development. And while we know that the synapse is extremely important for allowing neurons to communicate, we also know that synapses aren't static structures, and the way that they form and function changes throughout our lifetime. So shown here is just a plot thinking about synapse number versus age. And we know that during development, there's this intense period of synapse formation. There's actually an overproduction of synapses that are then eliminated during adolescence to get the correct circuitry. Throughout most of our lifetime in many brain regions, these synapses will be stabilized in their mature state. And then as we enter aging, even in the absence of any neurodegeneration, you can begin to see some loss of synapse function and even loss of connections in some brain regions. So we think it's important to understand in a typical brain how these circuits form and function. Importantly, if we think about many different neurological disorders that also appear at these different life stages listed here, we also know that one of the first things that can be seen is actually a change in the function of at the level of the synapse. So again, we think if we can understand what regulates these connections at these specific life stages in the healthy situation, will give us some knowledge of how we could potentially come up with new approaches to protect these synapses across these different disorders. So the way that my lab thinks about this, as was mentioned in the introduction, is to consider all the cell types present in the brain, so the environment that the neurons find themselves in. So here is just a cartoon of the neurons shown here in cyan, about half the cells in the brain, and then the glial cells interact with them and have many important functions in regulating neurons throughout life. And we're focused on the astrocytes, these green cells named for their star-like morphology. And research by many groups has shown that these cells have important functions depending on the specific life stage that we're thinking about. So what my lab has been interested in mainly until recently is their role in development, where it's been shown that they can support neuronal survival through production of trophic factors and also promote synapse formation and maturation through distinct secreted proteins and also membrane proteins. We know in the adult brain, they can have important roles in regulating ongoing synaptic transmission. So having, for example, uptake transporters for different neurotransmitters, and also providing metabolic support to the neurons in the form of lactate, but also lipids such as cholesterol, which are very important for synaptic function. Finally, if we begin to think about aging, at least at the transcriptional level, there are now numerous studies showing that these cells change their properties in ways that we predict may be damaging to neural circuits. So they seem to decrease their metabolic support, they downregulate this cholesterol support pathway, and they also upregulate many things we would normally think about in an inflammatory sense that could potentially be damaging for the neurons and the synapses. So together, we think studying these cells, particularly at the level of the synapse, is going to be important for thinking about their function in different neurological disorders. So that was just a cartoon image just to show you what one of these cells really looks like if you're not familiar. This is a dye-filled astrocyte actually taken from Mark Ellisman's lab here at UCSD. And you can see really the amazing morphology of this cell. This is the cell body, the major branches, and then the fine processes radiating out into the neuropil. And we know that through these fine processes, you can see this by electron microscopy here, these astrocyte processes actually interact with many of the neuronal synapses that are present, or in this case, this dendritic spine. And by having this close association, this is what allows them to have many of those properties or fulfill the properties that I just described to you about regulating synapses throughout life. And an important foundational piece of knowledge that's relevant to what I'll tell you about in the rest of the talk is that work done about 20 years ago showed that if you take neurons out of the brain or the retina and put them in culture, these neurons by themselves won't form synaptic connections with each other. And it's actually having astrocytes or the proteins astrocytes secrete into the extracellular space that tells the neurons to make these synaptic connections. So we know that by themselves, neurons can't carry out all the functions that we would like them to do. So, as I said, this area of how astrocytes regulate synapses is something my lab has been deeply invested in, and understanding what are the molecular mechanisms that are involved in this process. And here I'm giving you a timeline. This is based on mouse development timeline and looking at synapse formation in the cortex. So in the first couple of postnatal weeks in the mouse brain, this is an intense period of synapse formation. And we and others have identified different astrocyte-secreted proteins and also lipid cholesterol that are very important for regulating this initial synapse formation. We then, through further work, discovered that actually, as the brain matures, the properties of the astrocytes changes, and they begin to produce different factors shown here on the bottom, or actually kind of maintaining those synapses in place. Finally, as I mentioned, we and others have shown mainly through transcriptional studies that as the brain matures and goes into aging, the properties of the astrocytes changes, and they upregulate things that we would normally associate with the potential for synapse loss, such as complement proteins. And they also downregulate the ability to produce cholesterol, which we know in development is very important for supporting synapse function. So together, this makes us think, if we understand what's happening in the typical brain, can we now use this knowledge in different neurological disorders? And that's what I want to spend most of my time talking about, whether some of these things that we know are normally happening actually change in different neurological disorders. And if we correct those changes, can we have any benefit? So there's many reasons why you may be interested in this question. of why astrocytes and non-neuronal cells may be playing important roles in different neurological disorders that are associated with different life stages. And one of this graphic shown here from Kevin Kelly is just a nice way of thinking about, if we look, this is data from the human brain, and ask, if we look at which cell type expresses specific genes, and genes that are normally associated or changing in different neurological disorders, which cell type are these genes enriched in? And what this study showed essentially is, if we look across these different neurological disorders that strike at different life stages, anywhere you see kind of a darker colored box or a star, this is saying the genes associated or changing in that disorder are actually most enriched either in microglia, astrocytes, oligodendrocytes, or neurons. And what's striking when you look at this plot is that, particularly for astrocytes across the entire lifespan, many of the significant changes are actually happening in the astrocyte cells, more so even than neurons in some cases. So this is one reason why we think studying the non-neuronal cells is important in different neurological disorders, to try and understand what contribution they could be having. And due to our interest in development, the first study that we were looking at this was looking at astrocytes in the context of neurodevelopmental disorders. And we were really inspired to do this study based on many works in the literature, looking at how astrocytes could be impacting neuronal development in different genetic forms of neurodevelopmental disorders. So here I'm showing an example from Rett syndrome, which is caused by mutations in the MECP2 gene. And what the researchers did, this was using a mouse model, but it's been repeated using human cells, iPS astrocytes. They showed if you take wild-type neurons and grow them with factors secreted by wild-type astrocytes, which what we call astrocyte conditioned media, you get fairly elaborate dendritic outgrowth and complex dendrites forming in these neurons. If you take these same neurons that do not carry the Rett syndrome mutation, and you now grow them with astrocytes that do carry the Rett syndrome mutation, you now see an overall stunting of this dendrite outgrowth and less complexity. So this is really important study but that the astrocytes could actually be imparting some of the changes that are seen. And around the same time, or the next 2 years, other groups showed that this isn't something that's specific to Rett syndrome. This is also occurring in other disorders such as Fragile X syndrome and also Down syndrome. So we wanted to understand, a former graduate student, Alison Caldwell, wanted to ask, well, can we actually identify what's changing in these astrocytes across these different disorders, as the first question. And secondly, are there any commonalities we could identify across these 3 different disorders that could give us insight into some core changes that occur to astrocytes in these different contexts? So I'm just going to summarize the project for you because this has been published. But what Alison did was she took astrocytes from 3 different mouse models of these genetic neurodevelopmental disorders, so Rett syndrome, Fragile X syndrome, and Down syndrome, along with wild type typically developing. And she developed a culture model where she could grow these cells in isolation in culture in a defined serum-free media. And this is important, it's serum-free because we know that serum exposure of astrocytes makes them reactive, so it puts them in this disease state. And then she actually collected everything they release into the media, what we call astrocyte conditioned media, to perform mass spectrometry, and also collected the RNA to do RNA sequencing. And kind of the overview of the findings is that by doing these approaches, we can reliably detect around 1,000 proteins and around 12,000 genes. So we wanted to know, can we see any changes in the protein secretion? Because we know the effects that are being seen of the change in signaling between the astrocytes and the neurons is something happening at the level of protein secretion. So when Alison looked at this across all 3 different disorders, so this volcano plots on the top are looking at protein changes in the 3 different disorders, down-regulated proteins on this side, up-regulated. See in each disorder, many of them being upregulated, and that the number varies depending on the disorder that we're looking at, with the most proteins being altered actually in Down syndrome. What was surprising to us at the time, but has since kind of been shown by other groups and in other contexts, is if we look at the RNA sequencing data that we also generated from the same cells, we find while we also detect many differentially expressed genes that are occurring in these contexts, there's very little overlap between the protein secretion changes and the RNA changes that we see. And what this tells us, and has kind of motivated some of the studies I'll show you later, is that we really need to be studying astrocytes at many different levels to understand how they may be altering their communication with neurons in these different disorders. So as I mentioned, what Alison was very interested in was, can we actually identify any commonalities in how astrocytes change their protein secretion or change their communication with the neurons across these different disorders? So we looked at the overlap of the upregulated proteins and found actually there's a very high overlap. 88 of the proteins that are significantly upregulated are actually increased in all the different disorders. And shown here is a ranking of the top 15 of these, and these are ranked by their abundance in the wild type astrocytes. Yes?
Yeah, thanks. Just on that last, by finding the dissociation between what was secreted
Yep.
versus what was seen as upregulated,
Mhm.
does that mean that you're just upregulating genes that are increasing the secretion process or something?
Yeah, so that's one hypothesis that we're looking into. So it's a few things. It could be everything's happening post transcriptionally. So there's a different level of regulation that we should be looking at, what stimulates the secretion, for example. So those are genes that we're interested in. The other thing is we're kind of related to that. By looking at the secreted proteins, we're very far from transcription. So I'll show you some data later where we've done similar experiments with Alzheimer's astrocytes and iPS models, where we also looked at cellular proteins, so the lysate. And there you do see more concordance between the RNA and the protein. So I think it's the further away you get from that, the difference you see. Yeah. Yep. So in terms of these top proteins, there are many that are interesting that we're following up on in the lab. But the one that Alison chose is actually the top one on the list, this protein called IGF-BP2. And this stands for insulin-like growth factor binding protein 2. And we were interested in this because, as I said, it goes up across all disorders. The canonical role for this protein is as a secreted inhibitor of IGF signaling. And we know in development and also neurodevelopment, like IGF is very important in typical development, and in many neurodevelopmental disorders, there is deficient IGF signaling. So we hypothesized that perhaps one reason for the deficient IGF signaling because is you have too much of this inhibitor being released by the astrocytes. And Alison showed in cell culture that if she just adds excess of this protein to neurons, this is actually sufficient to inhibit neurite outgrowth. And that if we have a blocking antibody that blocks the ability of the binding protein to bind IGF, this rescues this effect, showing the functionality. So she wanted to know in an in vivo context whether this has any benefit. So she took the mouse model of Rett syndrome, and she looked at one of the phenotypes that has been detected in this early age, so the first postnatal week of development, which is actually a decrease in the soma size of the neurons in layer 5, so the deep layer neurons. And this also correlates with a decrease in the size of the dendritic arbor, but the soma size is kind of a more reliable readout that we can look at experimentally. So Alison first established that this is happening. So she compared wild type and Rett syndrome mice, looking at the size of the neuronal soma by labeling the neurons with GFP. And then she asked if we look in the Rett syndrome condition and inject either a control antibody or the blocking antibody against IGF binding protein 2, we see any increase in neuronal soma size. And this is indeed what she did see. When we quantify this in deep layer neurons, so layer 5, averaged by mouse or across all cells that she looked at, we do indeed see a small but significant increase in the size of the neuronal soma when we introduce this blocking antibody during early development. And one thing that was important that she found is that if she looked in neurons in the upper layer, so layer 2, 3, if we compare the wild type and the Rett, they don't intrinsically show a difference. And when she introduces the blocking antibody, it has no effect. So it's not that we're just causing a general overgrowth or increase in size, but it's really correcting a deficit that's present in this specific class of neurons. So this is just one of the candidates that we followed up on, but this has led to many different ongoing studies that we have in the lab. And kind of leading into what I'm going to tell you about today, one of the things we noticed in our mass spectrometry is that a number of the proteins that are altered, shown here in bold, are things that we would typically think of are associated with Alzheimer's disease. I guess Mark Ellisman has a question.
Uh-oh.
Hi, Mark.
Hi, can you hear me?
Yep.
I just wonder, you identified IGF-2 as a key change.
Yep.
And that's going to influence OXPHOS versus glycolysis significantly. And the terminals need a lot of mitochondrial vitality.
Uh-huh.
You're analyzing protein, but I wondered if you looked at RNA production for those kind of bifunctional pathways, because in Alzheimer's, you shift to glycolysis.
Yeah, that's a great question. That's not something that we followed up on the mechanism in the first study. As I'll mention at the end, we are now looking at this protein as well in the context of Alzheimer's models. So I have a postdoc, Tao looking at this. So that's a great point, something that he can be thinking about when looking at the mechanism to also be looking at energy production as well.
Yeah,
So
we can follow up. That's great. Thanks.
thanks. Okay. Yeah. So as I say, based on this overlap, it got us into the Alzheimer's, which I'll get to in a minute. And it's not just the upregulated proteins shown here. There's also downregulated proteins, excuse me, such as pleiotrophin, which a postdoc in my lab, Ashley Brandenborough, who you may know as part of the ADRC, has been working on. And she has a paper back in revision asking whether actually restoring this downregulation of pleiotrophin in a Down syndrome model is beneficial in rescuing some of the dendritic and synaptic phenotypes that are seen. And also a former student in my lab, MD PhD student, James Deng, who's now back in the clinical phase of the program. He showed that one of the other pathways that we found is altered, which is actually an upregulation of BMP signaling in Fragile X syndrome. When he corrects this in the mouse model of Fragile X, this also has some benefits in terms of behavior and synaptic number and function. So this is how we, I would say much of what we focus on in the past in the lab was really trying to understand how astrocytes function in development and could be impacted in neurodevelopmental disorders. But based on these findings a number of years ago, we got interested in thinking about broadening out into other stages of life and other neurological disorders where astrocytes have been implicated to see if we could make any progress in these areas. And of relevance, obviously, for all of you, the main disorder we've been focusing on in neurodegeneration is looking at alterations to astrocytes in the context of Alzheimer's disease. And we really are very much interested in thinking about these early stages of neurodegeneration where you see a change in synapse function and synapse number before you see gross pathology accumulating. So we wanted to understand whether across the kind of progression of pathology, whether we could identify how both the astrocyte transcriptome and proteome are changing, both in vivo and in vitro, and whether there are targets we could go into block to see if we can change synaptic function. And as I say, I've been looking at this both in typical aging, but also in Alzheimer's disease models. And the hypothesis we came in with was if we can identify how the astrocytes are changing, particularly at these early stages, if we block these changes from happening, could we actually have some benefit on the synapses and the circuits and the behavior, and maybe even slow down the progression of the disorder? The opposite approach, which I'll just briefly mention at the end, is being agnostic to how an astrocyte changes its state across the Alzheimer's disease progression, but asking, given our knowledge, what we've learned about what these cells do in the healthy brain in typical development, can we re-express these cues exogenously in a way that would be actually beneficial to synapses to promote their stability or prevent their loss, and see if that will have any benefit. So most of what I want to tell you about now is work by a postdoc in my lab, Isabel Salas, who's really been leading our work to understand how astrocyte-synapse interaction is altered in Alzheimer's models and really focusing on this at the RNA level. And I should say that Isabel recently posted this on bioRxiv if you're interested to kind of see more details of the work that she's been doing. So the way that we did this in this project was to use mouse models that show, oops, no, I didn't. I'm just going to introduce Alzheimer's to people who don't need to introduce. But just to say, thinking about, see, I forgot what I was doing. Thinking about like canonical features, late stage amyloid plaques and tangles, but we were kind of interested in these early stages that could be seen in changes in neuronal function, particularly with mild cognitive impairment, where you can see perhaps an increase in neuronal function before you get into a decrease in neuronal function. So trying to understand the place that we could do something beneficial. And the way we did this was, firstly, we're using this, in this project, we're looking at mouse models, and we're focusing on astrocytes in hippocampus, a brain region that's been shown to be impacted in the disorders, in the models that I'll show you. And the way that we're looking specifically at astrocyte changes is to use this backtrap mouse model, which is essentially, this is a genetic mouse model where all of the astrocytes in the mouse brain express ribosomes that have a GFP tag on them. So what this then allows you to do is to dissociate the whole tissue, use an antibody against GFP to pull out the ribosome and the mRNA, and then carry out RNA sequencing just of the astrocyte mRNA. And this is just to show you that if we look at enrichment for genes in the astrocyte pull down versus the total hippocampus, the kind of more orange colors show this is an upregulation of the gene. And you can see for astrocyte genes on the top row here, we've enriched for them, whereas we've had a depletion of all the other cell types shown here. So this technique works. So the two models that we've looked at is to ask whether we see changes to astrocytes and how they develop across time in a model of amyloidosis, so the APP-SWE, PS1 delta 9 mouse model, or in a model of tauopathy, so the tau P30S. And just to show, we also looked in our colony, in our mouse model, because things can be different across different institutions and genetic backgrounds of the mice. When biology develops, so we can see at 4 months, we see no evidence of plaques, but we do by 6 months start to see plaques developing, shown in red here, and many plaques by 12 months of age. And if we look at staining for GFAP, which is a marker of reactive astrocytes, this is also increased across the pathology progression and is strongest when we see the most plaques, which is what we would expect and many other people have published. We can do the same thing in the tauopathy model and look for 88 to show a hyperphosphorylated tau. And again, 4 months, not much happening. We have a big change by 12 months. And this is also when the astrocytes show really profound changes in their state. So using these mouse models, Isabel has done a large amount of RNA sequencing, looking at these three different time points, 4 months, 6 months, and 12 months. And also looked separately at male mice and female mice to ask if we see any differences, any sex differences in how the gene expression changes in the astrocytes in these different contexts. And today, I'm just going to show you the combined data where we combine the male and female data, but in the manuscript on bioRxiv, Isabel has identified some sex differences, particularly in the APP model. And what we can do is then just ask, if we just look at the total number of differentially expressed genes, how many are there at the different stages? And what we can see is actually early on, because we're looking here at bulk sequencing or the astrocytes combined, not many changes are happening when we have the significance criteria. But by late pathology, when you saw the astrocyte state change, what pathways are changing. And as one might expect from studies recently published in the literature and also from human patient studies, most of the upregulated pathways in both APP and tau are related to inflammation, interferon signaling, upregulation of complement cascade, for aging. So what we see in the downregulated pathways in the Alzheimer's models is this downregulation of things related to cholesterol biosynthesis, and also generally things that we would consider things that are positive for synapses. So this downregulation of synaptic support. Then, as with the developmental disorder work that I showed you, we wanted to ask, well, are these changes completely different depending on the type of pathology that's present, or do we see commonalities in changes? And what we can observe actually in the upregulated genes, there's a very high overlap between the APP and the tau condition, and most of the genes changed in the APP are changing in the tau, whereas in the downregulated genes, the tau have many more genes to begin with, but again, they're highly upregulated, overlapping rather. And in terms of what these changes are, as I just showed you in the previous slide, the upregulation is very much immune, and the downregulation is related to neurons. And yes, Mark. Hi, Mark.
Yeah. Complex 1 went down, right? Real oxphos, right?
You're going to get me working on mitochondria at this rate. Yep.
Well,
thank you for pointing that out.
No, what goes down is interesting early, particularly because you're losing oxphos support. But the mitochondria are in the presynaptic terminals, few in the astrocyte, fine vellus processes. So my difficulty is that the astrocytes are transferring oxphos mRNA to the presynaptic terminals. Your data is not inconsistent with that. Do you wonder why oxphos machinery is so abundant in the astrocyte but decreasing?
I mean, I'm not surprised it's abundant in the astrocytes because recent studies have shown astrocytes do have many mitochondria and also use oxidative phosphorylation. it's no longer thought they only perform glycolysis, which was a hypothesis for a while. So I'm not surprised by that. The fact it's going down, I think there's a general downregulation in other pathways I'm not showing on here of things that would normally be producing energy. So I think that's an important change that we should be considering, and whether that's cause or effect. If it's because the neurons are less active, the astrocytes are providing less energy production support, or if it's because the astrocytes stop doing this, that the neurons become less active. And I think that's an important question that we should look at in the future.
Yeah, I can see you're a fan of the cholesterol transfer. And
Yes.
we know there's spinules that go from the astrocyte to the presynaptic terminal, probably ferrying that.
Yeah, I mean, we're thinking about
There's no reason to believe the astrocytes aren't also supplying messenger RNA.
Sure, it's just not something that we could look at in this data, because this data is just the astrocyte RNA sequencing, but definitely the whole energy, something we should be bearing in mind.
Yeah. Anyway, I just wanted to flag it because there's something we don't know, but your data suggests something perhaps.
No, as I say, very excited to get other people's input on the data. It's a big RNA sequencing data set, and to have future collaborations. So thank you.
Bye.
Okay, so as you can see, many changes, many things Isabel could work on. But what we focused on in this project is the main interest of the lab in asking how many of these different astrocyte secreted proteins or lipids that we know in the developing brain can have an important support function at the synapse, or in increasing synapse number, or changing recruitment of AMPA receptors to synapses, for example, whether these are altered across the progression in the Alzheimer's models. And when we look at this across both the APP mice and the tau mice, so shown here is the 4-month, 6-month, and 12-month, and we're looking at the log2 fold change of the disease model compared to wild type. Blue is a downregulation, red is an upregulation, and star means significantly different. And what you can observe is that generally, if we look at genes that would normally be involved in synapse elimination processes, such as the complement proteins, these are mostly upregulated across disease course. The exception is MERTK, which is actually involved in phagocytosis of synapses, and this gets downregulated in both disorders. Whereas if we look at genes expressing factors that would normally be promoting synapse number and synapse function, generally we see a downregulation of these, with the exception of thrombosbondin 2. And for us, one that caught our eye is this gene GPC5 encoding this protein glypican-5. And we were interested in this because it goes down significantly in both of the models that we're looking at. And while it's only significant at 12 months, this is something that's beginning to go down already at 6 months in the APP mice. So let me tell you what glypican-5 is. This is a GPI-anchored heparan sulfate proteoglycan. So this is the general structure. This is a protein that's actually present on the surface of the membrane of cells, and it can exist in this membrane-anchored form, or there are endogenous proteases that cleave it, which make a secreted form which is released into the extracellular space. And we have previously shown that other family members, particularly 4 and 6, when produced by astrocytes, have important roles in the developing brain in inducing synapse formation. And this can be when it's in the kind of this secreted shed form. And other labs have shown important roles actually for neuronal forms of these proteins as well in regulating synapses. And we've been interested in this protein glypican-5 for a while, because this is something that's enriched in glial cells in the brain, particularly astrocytes and OPCs. And we have a paper in revision where we've actually shown what happens when you get rid of this protein just in astrocytes in the developing brain. And essentially what I'm showing you here is immunostaining of synapses. So this is in the cortex, this is intracortical synapses, showing when we remove glypican-5 just from astrocytes at 1 month of age, we see a significant decrease in the number of synapses that are present, particularly a decrease in the GluA2 AMPA receptor, which is associated with mature synapses. And we also see a significant decrease in the size of the presynaptic boutons, particularly at the thalamocortical synapses, which are usually these larger terminals with electron microscopy. So we know in the healthy brain, in the adult, this is something that's important for synapse function and synapse stability. The other reason we are very interested in this gene is because of all the amazing studies that have come out recently, particularly single-cell sequencing studies from patients, post-mortem samples with Alzheimer's disease, which have shown consistently that in astrocyte fractions across these patient samples, that there's a significant downregulation of glypican-5 at the mRNA level. And this isn't something that is just happening in Alzheimer's disease. There are studies also looking in frontotemporal dementia and multiple sclerosis where people have shown the same thing. Specifically in the astrocyte fraction, you have this significant downregulation of glypican-5 that's occurring. And another thing that's important to note is that this downregulation of glypican-5 is actually anti-correlated with GFAP. So as the astrocytes become more reactive and upregulate GFAP, you get this concurrent downregulation of glypican-5. So very interested in whether this downregulation is something that could be impacting synapses and circuits. And Isabel also validated this, that this is happening in the APP model, shown here using in situ hybridization. So this is in situ hybridization for glypican-5 in the hippocampus. This is a zoom in, and then showing staining for astrocytes with S100 beta, and then for amyloid plaques with the 6E10 antibody. And what she observed is that really those astrocytes away from the plaques actually have maintained expression of glypican-5, but those astrocytes that are close to plaques or touching them are really downregulating this glypican-5, suggesting it's something about this reactive state of astrocytes around the plaque that's really downregulating this factor. So with all of this evidence in hand and seeing that something's also happening in patient populations, Isabel asked the question of, well, can we actually stop this downregulation and ask if it does anything beneficial in the context of the Alzheimer's mouse models that we're looking at, particularly the APP model. And the way that we're doing this in the lab and a lot of our experiments is to make use of these AAV PHP EB viruses. These are viruses that you can give peripherally, so into the bloodstream. So we give retroorbital delivery, and they actually cross the blood-brain barrier into the brain, and we can target them to astrocytes by using this minimal GFAP promoter, so that the cargo that we deliver will be specifically overexpressed in astrocytes and not in neurons. And this is shown here in this image and this quantification that we can overexpress either glypican-5, and we put this HA tag on it so we can identify where it is, or using this control SMFP protein, which also has HA tags, we can target about 70% of astrocytes in the hippocampus with overexpression of either glypican-5 or this control protein. And we can then ask, looking in this APP mouse model, and we're looking at both males and females here, we can ask whether overexpressing this factor early, so before it's normally downregulated, has any benefit on synapse function or animal behavior or synapse number. And the two different time points that we've looked at, so we're overexpressing this factor at 2 months of age. We chose this based on data I'm not showing you here, where we actually looked at neuronal function in the hippocampus. So we can make ex vivo slices shown here and record from the neurons using whole cell patch clamp. And so we're recording from CA1 pyramidal neurons. And what we found is at 4 months of age, we actually see a significant synaptic hyperactivity happening in these neurons. So we see more spontaneous synaptic events. If we look at 2 months of age, this hasn't appeared yet, whereas if we look at 6 months of age, this has now switched to a hypoactivity. So these neurons are actually significantly less active than the control. So we asked whether, given we know that glypican-5 can impact synapse stability and maturation, if it could prevent this hyperactivity from developing at 4 months of age. And we also asked at 6 to 7 months of age, where you begin to see very subtle changes in behavior in these mice, which I'll show you whether this has any impact here. So first, in terms of the electrophysiology, so what we looked at here, these are recordings of spontaneous excitatory postsynaptic currents from CA1 neurons at 4 months of age. So this is the wild type with the control protein, the APP with the control protein. And as you can see in this quantification here, there's a significant increase in the frequency of synaptic events detected. It's actually about a doubling. Importantly, if we now look in the groups that have been overexpressing the glypican-5, so the APP mice, we now don't see this happening. So if we compare the APP control to the APP glypican-5, we now see that there's a significant decrease in the number of synaptic events detected, and this is no longer any different from the wild type condition, suggesting that by overexpressing this factor in astrocytes before this phenotype normally develops, we've either prevented it from happening or slowed it from appearing. In this mouse model. So this is kind of a standard model asking over a period of training days whether the mouse here can actually, when it's put in this arena, can it locate the escape box over here based on spatial cues. So we do this training over a number of days. We then test, and then we do a reversal training where we put the escape box on the opposite side and ask, can the mouse learn where the new escape box is? And again, we do a probe. As I mentioned, at this age, the phenotypes are very small, very subtle in this mouse model. So I'm just going to show you the learning data. Essentially, what we found is if we look at the learning across days, so this is each trial per day, trial is 2 trials per day. In the learning phase and the reversal phase, you see there's very little difference between these. Protein, or red is APP with a control protein, and the dashed lines is overexpressing glypican-5. And actually, the main difference that we found is that in both of the groups overexpressing the glypican-5, both the wild type and the APP, we have this enhancement of learning rate. So over the first few days of the test, they learn the task more rapidly, but by the end of the training, everyone gets there. So when we do the probe trial, we also see no differences, all the mice are able to learn age. This is really something happening very on in this, in the at this stage, any changes in synapse composition. At age again, there are fractal changes that are happening. The increase in terminals in the hippocampus. We see that in these mice. We do not see any impact of glypican-5. What we do see is an impact specifically on GLUA2 AMPA receptors shown here in the CA3 region of hippocampus. Again, we don't see a genotype difference at this age. If we go later, we do see a genotype difference. But what we see is overexpressing glypican-5 is sufficient to increase this GLUA2 AMPA receptor, suggesting perhaps increasing these AMPA receptors has had some impact on the ability of these mice to learn more quickly in both conditions. And we, of course, looked at plaques. We saw no difference at this stage when there are very few plaques to begin with. We saw no impact of glypican-5 on the amyloid plaque load. And this kind of, we looked at this kind of for aficionados because this is a glycoprotein. There's a lot of interest or impact of glycoproteins accumulating in plaques or perhaps being involved in these things. So to summarize what I told you about going through this study, what we found is that overexpressing this factor glypican-5, which is downregulated with disease progression in these Alzheimer's models, does have some benefit in this synaptic function. It appears to stabilize or decrease the presynaptic hyperactivity that's normally occurring, and it's able to increase postsynaptic levels of GLUA2, which may have some impact then on the behavior that's seen. And obviously, you would like to know what happens if we go longer, when we see more profound deficits happening. Do we see this prolonged ability of glypican-5 to be protective? And we're also interested in other disorders because, as we said, pretty much every neurodegenerative disorder where people have published these sequencing studies, this goes down. So we want to know what it may be doing. So this is kind of to summarize this idea. As I said, we wanted to go all the way from looking at transcriptional changes, identifying candidates that we could work on, and also re-expressing these other synaptogenic factors. So in my last few minutes, I just want to briefly tell you about some other projects in the lab that we have. I just told you about Isabel's work looking at glypican-5 and astrocytes in Alzheimer's. Adrian Pommier is a postdoc who has found that in addition to this neuronal hyperactivity in early stages, you also see astrocyte hyperactivity. So an increase in astrocyte calcium signaling. So he's asking what happens if he blocks that. Does it have any benefit? Axinia Derivianko is a postdoc who's asking whether, like I mentioned earlier, if we overexpress cordon-like 1, which doesn't change in Alzheimer's, but normally is important for regulating synapses in the healthy brain, does this have any impact on the synapses that are present? And then Tao Tao is the postdoc who's following up on this question of asking whether if we block IGF binding protein 2 upregulation, which also happens in the Alzheimer's models, does this have any benefit like we showed in the developmental disorders? So quickly going to mention 2 projects in more detail so you can get an idea of what we're doing. So Ashley Brandenburg, who I mentioned earlier, postdoc in the lab, one of her projects has been looking at pleiotrophin changes in Alzheimer's disease, but she's also been leading our work looking at how the proteome changes in astrocytes in vivo in Alzheimer's disease. And the way that we're doing this is, again, using a viral approach to overexpress an enzyme, specifically in astrocytes. So this is this enzyme TurboID. And we put this enzyme TurboID present in the endoplasmic reticulum. And what happens is when you give exogenous biotin to the animals, basically any in the tissue and know that this came from an astrocyte and it was in the secretory pathway. And we validated this in a preprint that we have on bioRxiv led by James Deng, where he did this in the Fragile X syndrome mouse model. And he actually identified a number of proteins that are downregulated or upregulated in vivo in the astrocytes in Fragile X syndrome. So Ashley took this same approach and used it in the Alzheimer's models, again, overexpressing at these different time points I showed earlier, 4 months, 6 months, in this case, 9 months in the APP model and the tau model. And she found, indeed, if we look at the male and female mice, either in the APP or the tau, if we look at the differentially expressed proteins now, not genes, she can find many differences occurring, more in the tau than the APP, like we saw with the RNA as well. And also, interestingly, at this stage, not much overlap. So at least in the proteome, it does seem to be more disorder or pathology specific. And these are just some of the examples of the proteins here. So this is kind of fairly new data. So we're combing through it. Ashley's looking at it to think about what are promising things that we could follow up on to try and intervene. The other project I just want to briefly introduce is being led by Julie-Beth Bargado, a graduate student in the lab. And this is where we've been looking in the context of actually in human astrocytes here, so collaborating with Rusty Gage at Salk to ask how iPS cell-derived astrocytes from patients with sporadic Alzheimer's disease, if they are altered in terms of their function, their protein production, or their RNA. And this is the original cohort that we worked on, showing the designation of the patients. We've now expanded this to include some UCSD cohorts as well.
That hurt.
It's big enough.
Grant number.
It now has UCSD.
Okay.
It's now larger. This is what we started with. And we can see across APOE genotypes, across polygenic risk score, etc. And the main point that I want to make from this is that even though these are from patients with sporadic late onset Alzheimer's disease, we can still detect differences in terms of things that are upregulated or downregulated compared to the control patients. So we can see changes in the RNA with the gene expression, changes in the lysate proteins, the cellular proteins, and also the secreted proteins, both up and down regulated. And as I said, this was a fairly small cohort, so we've now included some from the UCSD Center for the RNA sequencing, and we're seeing similar patterns. And if we look in terms of the pathways that are altered, we can see different things if we look at the RNA or the protein. And in terms of the RNA, as we saw in the mouse models, a general decrease in things that would normally be supporting of synapses. And in terms of the lysate, we see things upregulated associated with DNA damage and downregulated again related to lipids. And I'm just going to finish, Mark, if that's okay, and we can do questions at the end. So just to summarize for you, just to give you an overview of what we're doing in the lab, really interested in understanding across these different life stages how our knowledge of astrocytes and the proteins they secrete could be beneficial in the context of different disorders in ways whether we could try and stimulate synaptic repair or prevent synapse dysfunction from appearing in the first place. And this is my group at Salk, lots of great people here. I showed you much of their work along the way, also lots of great collaborators, former lab members, and also funding. And I will finish now, and I guess we go straight to questions with Mark.
Yeah.